Hi, welcome to Kavitha's's NEET classes. In this lecture, we shall discuss a simple topic, the structure of DNA. So now many of you all might be wondering, what's so special about the structure of DNA? After all, we have studied it right from our school days. So we all know that it was James Watson and Francis Crick who proposed the double helical structure of DNA. And what are purines, what are pyrimidines, what is adenine, guanine? So what's so special about that? There's nothing special about the structure of DNA as such, but the approach which you have to follow for a competitive examination should be completely different from what you have been doing all these days preparing for a board examination. So be it this topic, that is the structure of DNA, or any other topic, you would have so far approached it with the point of writing a good answer in your board examination or your school exams, isn't it? You would have studied about what is the DNA, what is the structure, what are polynucleotide chains, what is the composition of the DNA. You would have learned how to make a diagram of the DNA and label what is a major groove, minor groove, and so on. But here, when you're preparing for a competitive examination like NEET, your approach should be different from that of what you've been doing or how you've been approaching your board examination. Here, you're not writing essay type answers. Say you're not writing a 5-mark answer on the structure of DNA or a 10-mark answer. Here, you have the answers in your question paper itself. So, this is an objective type question. This examination is all about thinking objectively. You are given a question followed by 4 different options, and you have to choose the best option or the right option for that particular question, and that too you have to do it within a limited time frame. So this is what makes the examination tough, isn't it? So when asked to write about the structure of DNA, all of you definitely can write a very good answer for 5 marks or 10 marks. But when given a question asking where is DNA present other than the nucleus in a prokaryotic cell or in a eukaryotic cell, and given 3 or 4 options, and when you read all these 3 or 4 options, you will have slight confusion thinking which one is the right one. So here, whenever you are preparing for a competitive examination, nobody is going to ask you how much marks or what percentage of marks you are scoring. So whether you score distinction or whether you're scoring first class, second class, or you're just having a just pass marks, that is not going to matter here. In a competitive examination, it is just like running a race. You have to outperform the other competitors who are also taking the examination. So here in a competitive examination scenario, you'll have to think objectively. Whenever you read any paragraph or any topic or even a single line, you need to analyze, not just understanding the concepts and memorizing few facts. Yes, definitely here and there, few facts like the name of the researchers or the person who discovered, few factual things we have to remember and we have to memorize. But in addition to that, we have to have a very clear understanding of the concepts. So conceptual clarity plays a very important role, and in addition to that, you should also analyze and you must be able to guess or make questions out of the topics whatever you are reading, isn't it? Say you are reading a paragraph, you must think like an examiner and try to frame questions out of that. You must try to frame questions thinking that you are an examiner and try to understand, okay, what kind of questions can an examiner make from this particular topic. So this will help you in approaching this exam objectively and score much better and achieve your goals, isn't it? So in future, we will be discussing more about the tips, tricks, how to approach various questions and so on. As far as this lecture, let us begin with the structure of DNA. So even before we start discussing about the structure of DNA, the components of the DNA, let's just go through the basics of where is the DNA present in the prokaryotic cell, eukaryotic cell, where it occurs, and so on. And then we should start discussing about the structure of DNA. The occurrence of DNA. Where does the DNA occur? So even before that, DNA. What is DNA? DNA is deoxyribonucleic acid, isn't it? DNA is deoxyribonucleic acid. So what is DNA? DNA is the genetic material which is responsible for transmission of genetic information across generations. DNA is the genetic material in most of the organisms. However, we also have certain organisms which have RNA as their genetic material. Say for example, the retroviruses. We have viruses which have RNA as the genetic material and they are called as retroviruses. So most of the organisms have DNA as the genetic material. The genes are present inside the DNA and it is the genes which are responsible for our characters or traits. Characters or traits can be very simple, like the shape of your eyes, the color of your eyes, the shape of your nose, ears, your skin color, hair texture. Any of these can be characters or traits. So your skin color, your eye color or the shape of your nose is determined by the genes which you inherited from your parents, isn't it? So whatever genes you have inherited from your father or mother determines your eye color or your eye shape or your nose shape or your skin color and so on. So it is the genes which are responsible for transmission of the information from one generation to the other generation. And the genes are contained in this genetic material called as DNA. Okay, that is deoxyribonucleic acid. The genetic material in most of the organisms is DNA. However, we do have certain organisms which have RNA as the genetic material. So what is RNA? RNA is the ribose nucleic acid. RNA is ribose nucleic acid. Where does the DNA occur in a cell? So we know we have two kinds of cell, the prokaryotic cell and the eukaryotic cell. The prokaryotic cell is the primitive cell which is present in lower organisms like certain bacteria, amoeba, euglena and so on. So prokaryotic cells are mostly found in primitive organisms, whereas the eukaryotic cells are the ones which are more advanced and it has well-defined organelles. So we shall be discussing more about the difference between the prokaryotic cell, eukaryotic cell, etc. in our future lectures. First of all, in a prokaryotic cell, the DNA is present as circular structures freely suspended in the cytoplasm. Here you can see this, isn't it? These coiled structures, circular coiled structures which occur freely in the cytoplasm in a prokaryotic cell. This is called as nucleoid. This is called as nucleoid or nucleoid DNA. In addition to this nucleoid DNA, we have circular DNA molecules which are capable of self-replication. These are called as the plasmid. Okay, so this is the nuclear DNA which is freely suspended in the cytoplasm and this is called as nucleoid DNA, and in addition to this we also have extranuclear DNA. Extranuclear DNA appearing as circular DNA molecules in the bacterial cell. These circular DNA molecules are capable of self-replication. That is, they can replicate on their own and they have been used as vector molecules in recombinant DNA technology or genetic engineering experiments. So how exactly this plasmid molecules are utilized in genetic engineering experiments? We shall discuss that in our future lectures. So, in addition to the nucleoid DNA, in a bacterial DNA, in a bacterial cell, you have circular DNA molecules which are capable of self-replication called as plasmids. Got it? Next, moving on to the eukaryote. The eukaryotic cells are present in higher organisms, isn't it? So all plants, animals and higher organisms have eukaryotic cells which have very well defined organelles. So how is the DNA present in a eukaryotic cell? In a eukaryotic cell, you can find that the DNA is present as a complex along with proteins called as histones. So you have a histone octamer. You have histone, 8 histone proteins which together form a histone octamer around which the DNA molecule is circled like this. So we get a chain of DNA with beads of histomers or beads of histone proteins. It appears just like this. You get histone proteins and it looks like a beaded chain. So this is how it is present in eukaryotic cells. What happens is this DNA along with the histone proteins is compacted in the form of chromosomes, and chromosomes are present inside the nucleus. Got it? So DNA along with the histone proteins are compacted to form chromosomes which are present inside the nucleus. So nucleus is a very well defined organelle which has a nuclear membrane consisting of nuclear pores, and in a eukaryotic cell all the other organelles also are very well defined. So it is an advanced form, isn't it? So in eukaryotic cell, in addition to the chromosomal DNA, do we have DNA in any other organelles? Yes. In addition to chromosomal DNA, in eukaryotic cells, you can find DNA in mitochondria. So mitochondria are also called as the powerhouses of the cell, isn't it? So this is the mitochondria. So in mitochondria also you can find extra chromosomal DNA. In addition to this, we can also find extra chromosomal DNA in plastids. Okay. So now that we discussed about the occurrence of DNA in a prokaryotic cell and an eukaryotic cell, now we must try to guess what kind of question can my examiner make from this, whatever I have studied. So say your examiner can come up with a question like this. So the question can be like this. What are the additional DNA other than the nucleoid DNA that is present in a prokaryotic cell? Okay. So what is the other additional, apart from nucleoid DNA, what are the other additional DNA that is present in a prokaryotic cell? So the question can be, what is the additional DNA that is present in a prokaryotic cell apart from this nucleoid DNA? And the answer options can be, first one can be plasmid, and the second option can be mitochondrial DNA, and the third option can be plastids, okay, and the fourth option can be all of the above. So now what will be the right answer? The question asks, what is the extra DNA that is present in a prokaryotic cell? So the extra DNA apart from this that is present in a prokaryotic cell is the plasmid DNA. So the right answer will be plasmid. Whereas suppose if the same question can be asked with respect to an eukaryotic cell. So if they ask what are the DNA apart from the chromosomal DNA that is present in a eukaryotic cell, and you have the same 4 options. So now what is the right answer? So instead of prokaryotic cell, if they are asking about extra chromosomal DNA that is present in a eukaryotic cell, so what will be the answer? It cannot be plasmid because plasmids are present only in prokaryotic cell. So here the right answer can be, say the 4th option is both 2 and 3. Both 2 and 3. That is, here we know that in a eukaryotic cell, extra chromosomal DNA is present in mitochondria. So 2 is the right answer. And it is also present in plastids. Right? So second one is also right and third one is also right. However, you can have only one right answer. So the 4th option, which includes both of them, will be the right option over here. Okay, so now are you able to understand how we have to think and formulate questions when we are studying any particular topic? This will help you a lot when you are facing the examination. So we shall further move on. Now that we finished discussion about the occurrence of DNA in a prokaryotic cell and a eukaryotic cell, now we shall discuss about the composition of DNA. What is the DNA composed of? DNA is composed of 3 components. Okay, DNA is composed of 3 components. First one is the pentose sugar. Second one is the phosphate group. And the third one is nitrogenous base. DNA is composed of 3 components. First one is the pentose sugar, second one is the phosphate group, and third one is the nitrogenous base. This is the phosphate group, isn't it? This is the phosphate group, and this is the deoxyribose sugar. So the sugar in DNA molecule is deoxyribose, whereas the sugar in a RNA molecule is ribose, isn't it? So RNA is called as ribonucleic acid and DNA is called as deoxyribonucleic acid, isn't it? So what is the difference between ribose and deoxyribose, isn't it? This can also appear as a question. So what is the difference between ribose, which is present in RNA, and deoxyribose, which is present in DNA? So here you can see that in the second carbon atom we have H, that is a single hydrogen molecule. That is why it is called as deoxyribose, whereas in a ribose sugar which is present in RNA, at the second carbon atom, instead of this we will have a hydroxyl group, that is OH. That is the difference between deoxyribose and ribose. Okay, at the second carbon atom if you have H, then it is deoxyribose, whereas if you have a hydroxyl group, that is OH in this position, it becomes a ribose sugar. So the DNA is composed of pentose sugar, phosphate group, and nitrogenous base. So what are the nitrogenous bases? We have 2 kinds of nitrogenous base. One is the purine and the other is pyrimidine. Purine are double ring structures Under purines, we have two types. One is adenine and the other one is guanine. So we have two purines. One is adenine, which is represented by the capital letter A, and guanine, which is represented by the capital letter G. And under pyrimidines, we have cytosine and thymidine. Cytosine and thymine. Sorry, it's not thymidine, it is thymine. So, under pyrimidines, we have cytosine and thymine. Pyrimidines are single ring structures, whereas purines are double ring structures. Okay, keep this also in mind. This can also appear as an option somewhere. So most of the time what happens is, you might not exactly know the right answer for a question, but based on your understanding of various concepts, you will at least be capable of eliminating the wrong options. So when you eliminate the wrong options, obviously you will land at the right option. So understanding each of these will definitely help you tackle such kind of questions. So purines are double ring structures. So purine are adenine and guanine, and we have pyrimidines which are single ring structures. That is, we have two pyrimidines, that is cytosine and thymine. So these are the four nitrogenous bases which are present in DNA, that is adenine, guanine, cytosine and thymine. In RNA, we have four nitrogenous bases: adenine, guanine, cytosine and uracil. Instead of thymine, so in DNA we have thymine, in RNA in place of thymine we have uracil. Okay. So did you understand so much? That is the components of DNA. We have three components of DNA. First is the pentose sugar, then the phosphate group and the nitrogenous base. There's no particular order for this. Okay. Next. So how are these three components of DNA, that is phosphate group, ribose sugar and nitrogenous base, linked with each other and arranged in the DNA? First, this is the deoxyribose sugar. The deoxyribose sugar is attached to the nitrogen base. This is the nitrogen base and this is the pentose sugar. Okay. The nitrogenous base and the pentose sugar are attached together by a glycosidic linkage. Shortly, I'll be telling you what a glycosidic linkage means. So, a sugar molecule is attached, a nitrogenous base is attached to the carbon 1 atom of the sugar molecule through a glycosidic linkage, and this combination of the nitrogenous base with the ribose sugar is called as a nucleoside. Okay, so I'll write it down here: nitrogenous ribose sugar plus nitrogenous base. The ribose sugar plus nitrogenous base is called as a nucleoside. Okay. So ribose sugar plus nitrogenous base is a nucleoside, and the nucleoside plus the phosphate group, okay, that is called as the nucleotide. Sorry, it's not phosphate, it's phosphorus. Okay, so keep this in mind always. The ribose sugar plus nitrogenous base gives a nucleoside. and nucleoside plus the phosphorus group gives the nucleotide. Okay. So here you can see very clearly, this is the nitrogenous base, this is the nitrogenous base and the pentose sugar. These two together form the nucleoside. Pentose sugar plus nitrogenous base form the nucleoside. To that, if you add the phosphate group, sorry, it is phosphate group. So to the nitrogenous base and the pentose sugar, which is called as a nucleoside, if you add the phosphate group, it becomes the nucleotide. Okay, ribose sugar plus nitrogenous base gives nucleoside. Nucleoside plus phosphate groups gives nucleotide. So did you understand what's the difference between a nucleoside and nucleotide now? So you can have questions making you confused between this also. So you might get a question asking, what are the components of a nucleoside? So the options might be, first option can be, it is the pentose sugar plus nitrogenous base. Okay, next option can be pentose sugar plus phosphate group, and the third option can be pentose sugar plus nitrogenous base plus phosphate group, and the fourth option can be all of the above. Okay, say the question is, what is a nucleoside or what are the components of a nucleoside? So in this case, if these are the four options, which one would you choose? So now you might get a confusion whether it is pentose sugar plus phosphate or pentose sugar plus nitrogenous base or all the three. So all these three, that is pentose sugar plus nitrogenous base plus phosphate group, is a nucleotide. It is a nucleotide. It is not a nucleoside. Okay, so if the question is which one is a nucleotide, then this will be the answer. Whereas if the question is what are the components of a nucleoside, then your answer should be pentose sugar plus nitrogenous base. Got it? So as in how you keep reading, you need to keep guessing questions like this and make yourself much more confident when you get the questions so that you can answer it. Fine. So now you know what is the difference between a nucleoside and a nucleotide.